A tailor-made production presents Viruses in the Environment. Viruses are the simplest organisms known to us in the universe. They're tiny, non-living particles that have, over the millennium, developed an amazing diversity. The complete virus particle is known as a virion, which comes in a wide array of shapes and sizes, ranging anywhere from 10 to 400 nanometers. The simplest of these virus particles is a membranous nucleocapsid, housing genetic information. Beyond this, there can be capsids, which have a range of features, such as for adhesion and movement of genomic information between the virus and a host, which allows for the propagation of viral genome. The life cycle of a virus is particularly unique. The virion must gain entry to the host cell to survive and reproduce. Once a virus enters a host cell, the nucleocapsid is shed or uncoated. The nucleic acid can hijack the host cell components in order to replicate and translate the genome. There are two common ways of this occurring, referred to as lysis and lysogeny. Lysis refers to the immediate employment of host proteins to replicate viral nucleic acids and produce viral proteins, which then result in the expulsion of new virions which can self-assemble in the host following production of component parts, generally leading to host cell breakage and death, hence lysis. The method of lysogeny is often seen in chronically infected virus species and involve integration of viral DNA and RNA into the host cell genome to continually produce and shed smaller numbers of daughter virions, allowing for survival of the host cell and therefore continued infection over long periods of time. This can be thought of as a more sustainable form of viral infection. Due to their amazing ubiquity, there are known relationships between viruses and all domains of life, including animal viruses, plant viruses, bacteriophages, and virophages. Bacteriophages, which are viruses of bacteria, have little motility, and as such rely on random encounter with bacteria to infect their one form of host. Employment of the lytic cycle in marine bacteriophages releases biologically important molecules from the host. The release of carbon and other nutrients from infected bacterial cells into surrounding environment is referred to as viral shunt, which allows for less competition in these areas. Release of sticky particles furthermore contribute to deep-sea ecosystems through the production of marine snow, which is a nutrient package which sediments down and feeds organisms in the aphotic zone. The alternative process, lysogeny, where a phage fails to lyse a bacterium following infection, can instead carry new genes into bacteria through transduction. This infection and subsequent incorporation of genomic information is called gene-mediated horizontal gene transfer, and accounts for the uptake of approximately 20 billion new genes in various bacteria per second. This occasionally results in significant alterations to the bacteria in question. For example, Prochlorococcus species, which have been observed to contain structural photosynthesis genes acquired through transduction. Through infection, bacteriophages causes a turnover of approximately 20% of all bacterial marine biomass per day, with an estimated of 10 to the 23 infections occurring per second. Scientists have found that some of these bacteriophages are actually tremendously large in comparison to other viruses. A new virus, known as the Mimi virus, was found to have a diameter of 750 nanometers, with a double-stranded genome of 1.2 million bases. Interestingly, this is larger than some bacterial species. When these giant viruses infect a host bacterium, they produce what is termed as a viral factory. These factories are built within the cytoplasmic membrane of their host, and it is where all the new virion particles are made using the host's own machinery. Studying these giant viruses, a new small type of parasitic virus was found replicating within the viral factory, preying on the virus and the host. These new viruses were labeled as virophages. The researchers found that these small virophages can only replicate within the host bacterium after the giant virus has infected it. As the virophage exploits its larger counterpart's hardware, it causes a reduction in viable virion production of the giant virus and reduces the amount of lysis that occurs in the bacterial hosts. A new type of virophage was found within a lake in the Antarctic region. This newly discovered virophage has a parasitic relationship with a larger Phycodna virus, which in turn infects groups of phototropic alga called praseopods. It was found that when the alga was infected with both the large Phycodna virus and the new virophage, it resulted in a 70% reduction in viable Phycodna virus particles. After extensive modeling of this three-way relationship, it was concluded that the virophage was causing a definite reduction in the time taken for algal population to reach high numbers again, which resulted in more frequent algal blooms. It is believed that these new virophages may actually make a huge contribution to maintaining a healthy microbial food web. Virophages therefore keep bacteriophages in check, which in turn control bacterial populations who form the basis of the marine food web. This has been a tailor-made production. Thanks for watching.